Assalamualaikum and good morning to everyone. Thank you, Ms. Safifi, for the introduction. And welcome to our first webinar series from sample to data, uncover the microbial diversity using Amplicon Sequence Analysis. So our webinar today will be talking about the Amplicon Sequence Analysis approach in studying microbes and environment. So if you have any question related to this webinar, feel free to type your question in the Q&A box. You can find the Q&A box on the toolbar on the top of this team application. So we'll try to answer the question, all the question at the end of this session. So as for today's webinar, I will be covering a few topics here. So the first one, I'll be talking about the microbes and metagenomic. And then next, we'll focus more onto the Amplicon sequence analysis. And next, we'll look into the Amplicon sequence analysis service offered by Apical Scientific and Berhad, where we look into the sample requirements such as the sample, the DNA and the DNA quality and and the DNA quality, and also we look into the demo report at the end of this presentation slide. All right. So first topic, the microbe and metagenomic. So a microbe or microorganism is a microscopic organism that's comprised either a single cell, unicellular, cell cluster, or multicellular, relatively a complex organism. These microbes can be found everywhere, whether in air, land, water, or even inside the animal guts. Microbes play very important roles in everyday life. For example, microbes are vital component of fertile soil, contains billions of cells in the soil. So it is estimated in a single gram of soil there are several billions of bacteria alone. Another example, in the human body, microbes make up the human microbiota, including essential gut flora. In our stomach contains billions of microbes which plays roles in food digestion. So changes in the gut microflora can affect human health. For example, several studies on animals shows that microbe profile in stomach may contribute to obesity. Microbe study can be used. Microbe studies can be used to, for example, to monitor the environmental health. For example, in the agricultural studies can be used to contribute to understand the better crop production. Other example, like in the food industry, can be used to monitor food processing to ensure the high quality product and the consistency of the products produced. Also, we can use to find a better enzyme to be used in industry or food production. So this is just a few examples of usage of microbes. There are many more that actually we can list. However, the most of these bacteria actually are yet to be cultured. So it is reported that less than 1% of these microbes can be cultured. So therefore, the culture approach is unable to accurately to represent the entire of the microbiota in the environment. Therefore, there's a need of different approach to study these unculturable microbes. So what is a metagenomic? Metagenomic is an approach to look into unculturable microbes. It started in around year 1980s. However, during that time, the approach more involved in the functional-based screening, not so much on the sequencing, because the sequencing is considered quite expensive. Using a single sequencing platform, the cost is very huge. So because you need to do the cloning and then you need to sequence fragment by fragment, so the cost is very huge. However, this approach, nevertheless, this approach enables us to build a better understanding about the microbes function and environment. However, due to the cost, due to the cost of the sequencing, and also due to the laborious, we need to have a trained personnel, and then need to have a machine that to maintain the library. So this the progress of this metagenomic is quite slow during that time. Metagenomic application on the NGS platform, so around year 2000. So introduction of NGS platform revolutionized the genetic studies. So this revolutionize also happened on the metagenomic. So this enable us to simplify the workflow, which also able to reduce the sequencing costs. So the sample can be processed without needing for the sample to be cloned, right? So from this DNA sample, we can process it, put in a library, a construct in the sequencing library, and then we can sequence them directly, right? So basically, there are two approach for metagenomic studies. From starting from the environmental samples, like for example, like soil, stool, water, and others. So the DNA were extracted and processed directly from the mixed microbial community. And this sample can be the DNA, the extracted DNA can be used for either in the Amplicon sequence analysis or in the shotgun metagenomic analysis. So what's the difference between the Amplicon sequence analysis and the DNA shotgun metagenomic analysis is that for Amplicon sequence analysis, you only amplify what we so call as a gene marker, which is present across the microbes. For example, we can use CVS, or for let's say for eukaryote, you can use either 18S or ITS to amplify this region, and this region can be analyzed to understand the abundance and also the taxonomy of microbes that present in the environment. While for the shotgun DNA metagenomic, we will shotgun, we have fragmentized the extracted DNA, and we're going to sequence them all from end to end. And using the bioinformatic analysis, we're going to reconstruct back this DNA metagenome. And from this analysis, we're able to understand the microbes, the taxonomy, and then also the function information that contains in the environment. So what's the difference between the Amplicon sequence analysis and also the DNA shotgun metagenomic? So first thing what I can say is the cost. The cost for the Amplicon sequence analysis is very cheap because you don't need to have a very high amount of sequence data for this analysis. So you just need around 100,000 of sequence stacks, sufficient enough to capture the microbial profile in the environment. For the natural gun metagenomic is considered quite expensive because you need to have a higher coverage, because you need to sequence all the fragments that present in the sample in the pool, and also the analysis take a lot of what they call the computer resources. So this added cost to this DNA shotgun metagenomic analysis. Apart from the cost and also the coverage, sequence coverage, the genetic material also for the we can look into the different into the genetic material that required for both approach. For Amplicon sequence analysis, we just need a low amount of DNA requirement, DNA amount, because we just need to PCR them. So we don't need to have a higher amount of DNA sample. While for DNA shotgun metagenomic, you need to have a higher amount of DNA. So we need to sequence because as I mentioned earlier, we have to sequence the whole DNA fragment. Right. For the taxonomy identification, limit to the gene target. So for example, if let's say your PCR, your gene target or your gene marker is focused on the CTS, Amplicon. So this CTS Amplicon will only cover the prokaryotes. It won't cover the eukaryote. So if you let's say you use the ITS or 18S, so you only cover the eukaryote alone. So you need to if let's say you want to cover more of this the taxonomy, so you have to use a multiple gene target to cover the microbes, the whole microbiota in the environment. While for DNA shotgun metagenomics, because you sequence the whole DNA fragment, so there's no limit to the taxa identification, right? And due to this reason also, you don't have the functional information because you only PCR the gene marker, so there's no function information we carry over compared to the DNA shotgun metagenomic, right? For the host contamination, because there's no issue handling with the host contamination because we just PCR the samples directly from the PCR. While for the DNA shotgun metagenomic, there's a challenge with the host derived contamination. For example, let's say you work with the animal guts. For example, you want to study the microbes in the animal guts. There's a challenge to ensure the purity of the sample, because if you have a lot more of these host derived contamination in the sample, then what you get is most of the data will become from the host rather than microbes, right? Right, since the Amplicon sequence analysis involve PCR, so there's a in PCR bias introduced, but however they have already optimized. This application has been optimized for many years, so we have reduced the PCR bias. So compared to earlier days, so PCR bias is an issue, but not so much up today, because we have studied and the application, the method has been well optimized. And on the DNA shotgun metagenomics, because there's no PCR involved, so there's no PCR bias in this for this DNA shotgun metagenomic analysis. All right. So for this for today's webinar, so I will more focus on the Amplicon sequence analysis. So for the DNA shotgun metagenomic analysis, so we'll talk perhaps in different webinar, or perhaps you have any question. You can just drop your question in our email, which I will share you at the end of this with this slide. You can share any question related to any of our service, and we try to answer all these questions later. All right. So for the Amplicon sequence analysis workflow, so basically the flow can be divided into three phases. So the first one will be the sample preparation. So starting with the mixed microbial community, so what we need to do is just we need to do the DNA extraction, ensure that we need to have a good quality and amount of sufficient amount of the DNA. Next, once it's passed, we can start with the second phase, which is the PCR. So we go to PCR according to the gene marker or the gene target, according to the customer need, right? And once done, we can start with the library construction and proceed with the sequencing. So at the end of this phase, we generate a sequence data. So using the bioinformatic tools, the sequence were analyzed. So we can cluster. First, we're going to cluster all these reads according to the sequence similarity and analyze them. So to match its taxonomy, and also to study the abundance of these microbes in the sample. So the usefulness of this analysis can be further expanded when you're working with the multiple groups. So for example, these multiple groups can be either from different location. Let's say we have water from water pond from different location. So you want to study the microbes between, let's say you give like a contaminated pond and an uncontaminated pond. So you want to study what's the microbe that's present in the environment. Or perhaps you would like to study the microbes at different time point. For example, that you are studying animal diet, want to study the different of microbes across time points, so you can collect the the sample at different time point. Or perhaps in different organ parts, yeah, and you want to study what are the microbes that is present in different location of any animal guts. Also, the multiple groups okay, this is just an example. So there are many types of groups. As long as they are different type of samples, we can use this approach to study, to understand the difference between one groups to another, right? Using the same workflow as I mentioned, right? So is using the same workflow, the sample will process through the sample through same process. So starting with the microbial community A, for example. So we will do the sample preparation. We're going to extract its DNA and ensure its quality. And once it's passed, so we can start with the PCR, library construction and sequencing. All this been done separately, right? And once we have the sequence data for each of these groups of sample, we can combine them during the BR bioinformatic analysis. In the BR analysis, apart from the taxonomy identification and abundance information, we can also compare the microbes abundance across all samples. Additionally, we can also provide more information such as the alpha and beta diversity, statistics analysis, and so on. Right. So just to remind that for the statistics analysis, multiple samples, to have some of this analysis, we need to have a replicate of samples so that we can able to do some of this statistic analysis. So at the end of this webinar, I will show you our demo report to show what are the deliverables that we can offer to you. Right. So Amplicon Sequence Analysis. So who can use this service? So you can, for example, if you are working with livestock or farming, you want to study the soil health, or you want to study the feedstock probiotic component, or you want to study what are the microbes that are present in the milk, right? So you can use this application to Amplicon Sequence Analysis to study the microbes present in the sample, or perhaps you want in the food industry. So it's kind of become more interest from this industry to understand, to ensure that the foods, for example, the prebiotic, like the Vitagen, like the Vitagen, the yogurt that we eat, they always have a consistent amount of the microbes, the type of microbe inside the food. So the Amplicon sequence analysis can be used to study the food, the component of the food. Also for wildlife, so we can use the stool from the animal stool to study the animal diet. In the health sector, for example, which is have gained more interest to study the gut health for healthy lifestyle, or even for the university or local research institute to do a fundamental microbe diversity study. Right. So this is just some of example. The lists are endless. So if you think that you want to study the difference of microbe diversity environment, so this application sequence analysis is one of the tools that you can use. All right. So now let's look into the sample requirement. So let's look into the Apical sequence analysis service offered and bioinformatics analysis offered by us. So first we look into the sample requirement. So we look into the sample requirement, the DNA quality and quantity. Next we look into the gene target, and also last we look into the sequencing coverage. Next we look into the bioinformatic analysis, and we look into the demo report that's delivered by Apical Scientific Sandia Mahat. All right. So yeah. So first the sample requirement. So for the synchron requirement, we don't really need to have a very high amount of DNA sample. So we just need to send us a DNA sample which amount of around 200 nanogram. The more the better, and the concentration should be around 10 nanogram per microliter, and volume must be more than 20 microliter. The most important part here is the quality. So the quality is you need to have around the OD260, 280, around 1.8 to 2.0, which no degradation and also no contamination. If you have difficulties on extracting the DNA, so we also offer DNA extraction service. We have provide this service to many of our customer, and so far it works well. So we can handle from stool, water, even soil sample. So we can handle DNA extraction service for many type of samples. All right. So next we look into the gene target. So the selection of a gene target is very important because it will determine which microgroup that will be targeted in the analysis. So for example, if you are working with CTS, so the target will be the prokaryote. Or if you're interested to study the fungi, so the ITS1 or ITS2 will be the suggest gene target, the gene marker. Or if you're working with the ATS V region, so you can also use eukaryote. Sorry, if you study the eukaryote, you can use the ATS V region as well, right? So apart from this, the gene target, we also can accept other gene marker, for example, like the CO1 genes or even tRNL. So as long the Amplicon sequence range is around 350 base pair to 500 base pair. Right. So because the size range limited here is because of Illumina platform. So the Illumina platform only accept a short fragment, DNA fragment. But we also, apart from the Illumina platform, we also offer uh, our nanopore sequencing platform service. So using the nanopore, we can sequence a larger fragment size. For example, For prokaryote, even rather than we target the V region, the what's the V region, or explain in the next slide. So rather than targeting the V region of a CTS, we can, in fact, we can sequence the whole CTS region starting from the 1 up to the 1.5 kb of this variable region. So this will have a better coverage of the microbes that's present in the environment. So the only the downside of this nanopore sequencing platform, just the cost will have a little bit slightly higher compared to the Illumina platform, right? Um, Yeah. So the next we look into continue with the gene target. As I mentioned earlier, because the Illumina platform can only target region no longer than 500 base pairs, so we unable to sequence the whole CTS region. So this is an example. So this is a CTS region. So basically, for the CTS, we have 9 variable regions starting from V1 to V9, right? Um, so um, so we can use a PCR primers to amplify a specific variable region. So different variable region will generate a different type of the result, different type of result. But basically, the V3, V4 are so far one of these widely used gene marker, gene target, which is have been shown by the Roche P et al. 2019. This is the mock data set. So using the V1, V3, and V4 basically can cover more or less the same as what is the mock sample have shown here, right? But nevertheless, some of other variable region are suitable at different. Some of these microbes are well target using different variable region. For example, the lactobacillus has been shown are better targeted using the V1, V2. So it depends on your study. So you have to look in the publication. So what are the gene target is suitable for your studies? But if you want to have a general overview of the microbes or in the environment, I can suggest that either V3, V4, or V4 will be one of the well studied variable region for this Amplicon sequence analysis. All right. And finally is the sample requirement, talking about the sequencing and sequencing coverage. So sample are processed and sequenced using our MySeq platform. So once the sample sent to us, so we do a sample QC and a PCR QC. So the customer will notify on these two QC before we proceed with the sequencing. So for sequencing, using a Illumina platform, so we sequence the sample using a 300 base pair per n, and the amount of data delivered is minimum of 200,000 raw reads or equal to 100,000 raw text, right? So yeah. So Apical Scientific Saidin Bahad offer a one-stop solution. So we can offer like sample preparation. So let's say we have difficulties on preparing the samples, so you can just send the raw material to us, like for example the stool, DNA, soil, water. We can process all the samples. So we will process the sample according to this phase. So for the sample prep, so we do the PCR, lab reconstruction, and sequencing, and finally we'll do a bioinformatic analysis to analyze the data. Right. So so far I'm glad to say that we have delivered hundreds of Amplicon sequencing analysis project and widely used target region such as the CTS, V3, V4, ITS, or the ITS or ATS V4 region. Apart from that, we also accept the cluster region as long as the size range within the 350 base pair to 500 base pair. Right. For the result deliverables, so the data will be analyzed using a KIME2 software, KIME2 tools, BI tools. So this KIME2 will analyze using the Amplicon sequence variable to approach. So the result will generate as an table generation. So we look into the result later when we look into our demo report. So from this table generation, we're able to look into the taxonomy identification and also the abundance. Apart from that, we'll use the data generated by the table, and then we'll provide in the figure form, for example, the bar chart, the corona chart, and the meta coder. We also provide the alpha and beta diversity analysis, and also the unified distance analysis, and UPGMA. We want to compare the samples variables, so to see the difference between one sample to another. We also provide the comparative analysis, for example, the Venn diagram. So we provide the Venn diagram. The customer can, when we when the customer submit the project to us, we provide a BI form so the customer can fill up in the BI form. So in that section, there's details for the Venn diagram. So it ups to the customer what are the sample comparison the customer would like to have. Okay. Apart from the Venn diagram, we also provide statistic between groups. For example, we provide a dissect2, NOC, and also LEFC. All right. So just important notes for the statistic between groups. We need to have at least we need to have two or more groups of samples, right? Because we need to compare the samples, so we need to have multi groups of sample, and each of the groups must have at least 3 replicates, because the statistics doesn't work well if you don't have replicates here. So we can recommend to have 3 replicates per sample to have a good statistics for the analysis, right? And also finally, for only applicable for the CNS, we also can provide the metagenome functional prediction using a PyCrust2. So this is a metagenome functional prediction. So we're able to find what are the gene that might be present in the PyCrust2. As I mentioned earlier, so the Amplicon sequence analysis, we don't really sequence the gene. So what this PyCrust2 is, they predict what are the functional information that might be present in the sample based on the taxa that's available in the sample, right? So later we'll go through what's the PyCrust2 result can offer when we look into the demo report. All right, so let's look into the demo report. Right. So once we complete the bioinformatic analysis, so the customer will receive a result like this. All the results are being kept in respective folders. So we have the Texa box plot, top 10, Corona chart, statistic analysis, and so on. And to view all this result in an efficient way, the customer can just click into this report.html. All the result from this folder will be linked into this report.html. You all right, so this is the our demo report HTML file. So basically, any result, any project, which doesn't matter whether CTS or 18S or fungi ITS, basically we deliver a same demo report. So you can see there's the link over here, the tab over here, where all the results are being kept according respectively to the result, to the analysis conducted. All right. So on the first page, we have the overview of this Amplicon sequence analysis. All right. So so the introduction. So what is the Amplicon sequence analysis about, and also the tools that we use. So how we analyze the data. So we have a brief introduction, brief explanation about the BI analysis conducted on the samples. And then next we have the overview, the BI workflow overview. So starting with the FASTQ file, so we do the quality assessment, so to ensure that the data are being delivered at a minimum amount of data promised. All right, and then once it's passed through, we will delivered the data in the data to pipeline, data to and KIME or KIME pipeline. So this pipeline will process will process where do the quality filtering and trimming, remove any chimeric, and finally we'll do the construct the ASV table. So based on this ASV table, you can identify, you have the abundance information as well the taxonomy identification for each of the ESV. So apart from this, you can also have a visualization. So we can see the taxa composition, the reflection curve, alpha and beta diversity, differential abundance analysis, unifrac, and many more. The bottom of this page as well, you can see the tools, the software that used for the statistic analysis. And lastly, you have the reference. You can use this reference for your write-up or for your publication or even for your thesis. All right. So next, we can look into the result, the deliverables from this Amplicon Sequence Analysis service. So I will just go through briefly on any of these the service, and we'll later at the end of this the slide will also provide to you the link where you can download this demo report and you can have the feel our demo report and see how useful of this our demo report for the Amplicon Sequence Analysis service. All right. So next we have the rate statistic. So the first page is a rate statistic. So this demo report contains 12 samples where we denote the sample based on the alphabet. So we have A, B up to L. So each of the sample contains multiple bars. So each bar depict the sample details. For example, the blue color, the details of the raw data, and then the filter rates, where we remove the adapter sequence. And the next will be the denoise F and denoise R, where the step where we do the quality checking, we remove any of the reads that have a low quality data. And next, once we do the quality checking on the forward and reverse reads, we do the merging. So any reads that cannot be merged, you have to exclude them, and we will take out only the merged data. So, and then next step, we do the chimeric check. So any data that contains a chimeric regions, we have to exclude them from our analysis. And finally, we do the rarefied rates, where we do a subsample across all the data, read subsampling across all data set to ensure that when we analyze the data, we look at the at equal amount of data set size. All right. So next is the taxonomy overview. So once the data are processed, the data will be clustered and analyzed from, right? So from this data, we'll generate the ASV table. Right, so this is the ASV table over here. So the first column here is the ASV ID generated by the data2 pipeline or the KIME2. So this is the sequence ID for each of the ASV sequence. So you can find this the sequence by clicking the download representative ASV sequence over here. Right, so for example, we want to find the ESV number 1 over here. So what you can do is you can just highlight this and then you do a copy, and then you go to the representative sequence .fast A, and then you do a search function and you highlight which are the sequence that relate to this ASV number 1. All right, so this is ASV number 1 and this is sequence. You can use this data for, let's say, you want to do a downstream analysis, want to do a blast, or want to design primers or whatever analysis you want to do from this data, so you can proceed using this information. All right. So apart from this ASV ID generated by the KIME2 or the data2 pipeline, we also provide a secondary ID. This is to simplify the ID generated by the original software. So this is more simplified, so easy to understand. All right. So this will be useful when we look into the Venn diagram. So because the Venn diagram, we have a very long list when we work with a large sample. So by simplifying the ID, we helps our customer to browse through the result. Right. The next column will be the sample column. right? So as I mentioned earlier, this demo report contains 12 samples, so depicted by the alphabet from A to L, and the number over here represent the abundance, number of sequence that can be found in respective samples. For example, here the ESV number 1, we can see that we can find there's a 45 number of sequence in the sample E and 177 in the sample H, but none in others. Yep. So we can show that the abundance information of this respective sequence in the samples. Apart from that, we can the data also will be matched to the database. All right, it depends on the what are the target region used. So for example, that this data has been analyzed using gene marker, the 16S gene marker. So we use a Silva database to do the taxonomy identification. So from this information, we can identify what are the species that might link to this each of the ESV. Right, so it depends on the gene marker that you use. So like for example, if you use a ITS for your study, then the Unite ITS we use it as a database to for the taxonomy identification. All right, so next move on to the taxa bug plot. So this is just based on the table, so consider as a raw data. So if you want to have the data in the figure format, so we provide a few result. For example, in the taxa bar plot, so we provide the result, we convert them in the bar plot. All right, so this bar plot, this is an active figure, so it's just not a plain PNG file. So you can move your mouse over the bar chart and to identify, to see what are the information that link in the respective samples. So for example, like if you move your mouse over this color, the green color, so you can see that this is this sample which is matched to the Licorio lactobacillus, and then the abundance is about 0.10. Okay, apart from that, so we also have the legend over here. So for example, that you want to find a specific microbes organism, so you can just double click on this any of the microbes over here, then they will highlight that only particular organism microbes, that particular microbes. So this will be helpful if you're working with microbes that are sample that are rich of microbes. So let's say we have hundreds of microbes identified. Using this function will help the customer. All right, so you can select any of the microbes of your interest, and then they will highlight the microbes that's present in the sample. All right, so by double clicking the legend, so we'll turn back to the original state. All right, so by default, when you click the taxa bar plot, the bar plot will be shown at the genus level, but we also provide the result at the different taxonomy level. For example, you would like to see the result in the phylum level, you can just click the link. You have a similar plot as what we've seen earlier. So you have a similar function, you can play around. Let's say you want to find the firmicutes, you can just highlight, double clicking at the firmicutes and then highlight the result. So this is very useful to our customer. All right, next we also provide the top 10 taxa based on the box plot result. So we provide across all taxa level. We have a phylum, class, order, family, genus, and up to species. Right. So this is based on the top 10 microbes that present across all samples. So we can see that we can do a direct comparison. What are the microbes groups that are widely present across all samples. All right, so this is top 10. Next, we have the heat map. We also, apart from the taxa bar plot, we can also provide the result based on the heat map. So we can identify the microbes abundance based on the color the heat map color. So the darker of the heat map color, the more abundance of the samples. The lighter the color, the lower of this the abundance of the microbes here. Right. Next, we have a Corona chart. So we also provide a Corona chart to show the abundance and also the taxa, the microbes that present in the samples. So each for Corona chart, each of these Corona chart circle here represent for one sample. So since that this is contains this demo report contain 12 samples, so you have a 12 circle Corona chart, right? And for Corona chart, help you to look into a specific microbe groups. For example, you want to look in details the firmicutes, for example. So you can just move your mouse over to this group and you click it and you click one more time. So the circle will just focus only on the firmicutes. So we can see what are the microbes abundance in the firmicutes. All right. And if you click a corona chart for other samples, so it will show at the same level. So it's, for example, like this is to have a highlight the firmicutes. So if you click the B also, they'll show the corona chart at the firmicutes level. All right. So if you want to use this data for publication, you can just right click and then you can save the image as a PNG and you can use them for publication. All right. All right. Next, we have distant matrix and also your PGMA. So this result can be used to identify, to compare the sample profile across all data set. So based on the taxonomy that's present. Right, so we provide either weighted unified and also the unweighted unified. Right, so the data based on the taxonomy that present in the sample. So you get there's score here that highlight the how close from one sample to another. Right. So there's a distant matrix and also UPGMA. So you might choose any of this result if you because some metrics are well performed on certain samples. So you can either choose a distant matrix or UPGMA to present your data. Right. Apart from that, you also have alpha diversity. So alpha diversity is a statistic to show um, the specific and the local scale. So want to see what are the information that carried by this the samples, respective samples. So there are a few alpha diversity alpha indexes that we provide. For example, we provide the observed, SHA1, ACE, Shannon, Simpson, and so on. So the details of this alpha diversity are been explained down here. All right. And at the bottom of this page, there's a reflection curve, so we can see whether if the sequence data able to capture most of the taxonomy present in the sample. As long as we can see that the graph, the reference curve reach a plateau, it will tell us that we have covered most of the microbes that present in the environment. Right. And next we have the ordination or the beta diversity. So we also provide a few indices. For example, we provide the PCOA, the CCA, RDA, MDS, and MNDS. So the ordination or beta diversity can give an overview, the difference between one sample to another. So we can see how this sample perform when we do a comparison. Right. Apart from that, we also provide the PCOA plot based on the 3D view, so we can play and we can move this plot to show to highlight or to present which the best for publication. So you can just move your mouse or you can just expand it and then you can just do a snapshot to capture this the PCOA plot here. Okay. All right. Next, there's a metacoder. So metacoder is a way of to do a comparative analysis. All right. So, for example, that this is the demo report that contains 12 samples. So basically, well, for demo reports, for this metacoder, you will have a two type of tree, the large tree where depict all these the taxonomy that's present in your sample. So you can see the taxonomy information starting from the bacteria up to the species information. And we have also the smaller tree here where depict the abundance information of this when you do for every time when you compare compare the sample. So there's for this comparison, you can see there's a two color over here, the brown and also the green. So anything that is highlighted, let's say highly highlighted in the green color, so means that they are highly abundance in this region, in this part of sample, where if it's highly, colored by the brown color means that they are highly abundant in this part of sample. All right. So for example, let's take one example that if you see the L versus A samples over here, we can see that this green color over here is which is highly present in A. So from there, when we match back to this large tree, we can say that samples that in the Firmicutes, Clostridia, or Bacilli groups over here are highly found in the A sample compared to the L sample. Right. So this is how we look into this metacodal result. Next, we have the DESEC non-parametric variable test and also LabC. So this is a statistic analysis that we can provide if you want to do a comparative analysis. Right. So for the DESEC, so we can see. Oh, before that, so for the DESEC, the comparative analysis is always a pairwise analysis. Is always one-to-one comparison. So if let's say your groups, you have more than two groups, for example, let's say you have three groups of samples, then the comparative can be done like group 1 versus group 2, group 1 versus group 3, and then group 2 versus group 3. So we can do that type of comparison. Right. So what you can get from this DESEC is the result over here. They highlight what are the microbes that is enriched in respective compared groups. So for example, that you can see that when we do a comparative across a group 1 and group 2, so we can see that the Selenomonaceae is highly enriched in group 2, and this column explain the fold change between this group 1 and group 2, and also the p-value to show the statistic information carried by this result over here. Right, another example over here, the Margonella, we can see that is highly enriched in group 1, and we can see there is highly enriched in group 1, so depicted by the negative number, and we also have the p-value provided in the result as well. Right, so this is example for DESEC analysis. So for the non-parametric variable test over here, we have the similar result. If you scroll down the mouse, so you can see the similar result as we see in this in the DESEC 2. So we have the microbes and also which group are enriched, and also the fold change, as well as statistics analysis. Right. So apart from this, the DESEC 2 and also the monoparametric multivariate test, we also have a LabC. So the LabC can be done in the compact groups. So if you have a multiple groups, all the groups are shown in a different colors. But for this the demo report, so there's only one groups enriched group, is group 4. So that's why I can't see the result for different groups. Okay, so there's only one group enriched, which is in the group 4. All right, so there's a statistic result. So DESEC, non-parametric variable test, and so LabC. So it depends on what result that might be suitable for you. You can choose either of these statistic result to present your data. Right, and next move on to the Venn diagram. Right, so as I mentioned earlier, so the Venn diagram is depend on the what customer requests. So the customer can fill up the comparison, the group comparison in the BI form, and we can deliver them. Right. So for example, like this is the Venn diagram where we compare all groups: the group 1, group 2, group 3, and group 4. Right. If you see that the numbers here represent what are the sequence, the ESV that present in respective sectors. If you move your mouse over the number or the sector over here, they will explain or the list of all the ESV that present in respective sectors, right? And to make things simplified, so we also provide an Excel file to depict all this number in sectors here. So by clicking this link over here and download. Right. All right. So, so the Excel file over here explain the sectors in this the Venn diagram. For example, just now we look at the group 4, so we can see that there's a group 4 over here, and then the number of ESV count here, a total of 26. And the next column will list out all the ESV sequence that present in the respective sectors. Right. If you like to know the details of this ESV, you can also do search function, and you can go back to the taxonomy overview data which seen earlier. We can do a search function by typing here, or we do a comparative search. Just Control C, and we'll highlight what are these ESV32 information here. All right, so we can see that for this ESV32, we've shown that it only found in this L sample, and is related to Paludibaculum organism. All right. Okay. All right. So that's example for the Venn diagram. Lastly, we move to the PyCrust tool. So as I mentioned in the slide, so the PyCrust tool is a way to do a prediction, functional information based on the microbes that's present in the sample, and is only available for the CTS data, because the PyCrust tool is well characterized for the CTS data, but not for other gene target. Okay, so for this PyCrust tool, we can see the abundance and also the function information. What are the enzyme that might possible present in the sample? Okay, so this is based on the prediction, based on the taxa that's present in the sample. For example, if you're working with the fermentation, for example, you expect that the microbes that present might carry that gene that might be involved in the fermentation. So you might see the gene that might present that relate to the anaerobic, or related to this fermentation will be listed in the list here. All right, so this is the result for the PyCrust. Right, so if you would like to have the details for the PyCrust, you can look into the folder, the PyCrust 2 result over here, and then you can click at this Excel file, and you have a more details result compared to the heat map we shown earlier. All right. So this PyCrust result explain about the based on the EC database. So the first column is the EC number, and then the second column will explain about the description of this enzyme, and the next column is the abundance information of the enzyme in a respective sample. All right, so that's about the PyCrust. And last page is about our company. So it's about our first base, and also we have our partners who help us in this bioinformatic analysis, which is a Nova LifeTech Private Limited. Okay, all right. So that's the last part of this demo report. Okay. So yes, if you have any inquiry on this NGS or any related service, you can email us at the ngs@apicalscientific.com. And yes, thank you very much for your attention. So we can look into the QA session.